So my name is Stephen Smith. I'm doing a PhD in Professor McCormick's lab here in Galway, and it's focused on using genomics to aid in conservation of the native Irish honeybee. So a bit of background about the situation in Ireland and why we're looking at conservation of the native Irish honeybee. This image on the right kind of sums it up. So this image shows the native ranges of each of the subspecies that the European honeybee is segregated into. And you can see that Ireland is part of the M lineage native range, so that's the subspecies that we have here in Ireland, the M lineage. And the conservation issue comes from the C lineage honeybees, so from Italy and Eastern Europe, coming into the M lineage range. So that's where the conservation risk comes from. So how are these honeybees getting here is the first question, and how much of them are getting here. So Ireland as an island, the conservation issue should be kind of solved for us due to natural reasons. We shouldn't be able to arrive here. So to find out how they're getting here and just how much of them are getting here, we did a survey of Irish beekeepers, just asking them, do you keep the native black bee that we have here? And around 6% of them said that they do not, so they keep a foreign subspecies. So that's the source of them. And honeybees, unlike other animals that you would import, they're very hard to contain. So if you were to import any other kind of animal, you can pen it in, contain what it breeds with, etc. Honeybees, the queen and the drones will fly vast distances to make it. So the area of impact of that animal that you imported is quite large. So that's why they're getting here. And we understand the biology and how they can go vast distances to mate. And is that happening, and can we detect it using genomics? So that's where my PhD kind of comes in, the background questions we kind of look at. So to solve this, we're using a pool-seq approach. So we go around Ireland, we sample 30 bees from each colony, we pool the DNA together, and then we work with the allele frequencies from that. Because the honeybee queen can mate with 15 different drones, this pool-seq approach is the most cost-effective way that we figured out to find out all that variation. So this is the workflow. The bioinformatics just kind of works at mostly allele frequencies and statistics. So first of all, I want to talk about how do we detect hybrid colonies. So we're going around Ireland, we're sampling 30 bees from each colony, that's our population. We want to know which is that population a hybrid. So we look at each colony as a population to see if it is hybridized. To do that, we use F statistics. So it's basically seeing if our test population C, is this an admixture event for populations A and B? And A and B here will be, A will be pure mellifera, so it's pure Irish colony. B will be a pure C lineage colony. So does C arise from a mixture event between these two? I'll show, so a negative result will show that admixture is present. So we do this for about 500,000 SNPs, but I'll show one SNP that has kind of shows you how this works. So if our Irish sample has an allele frequency of 0 at this SNP, our C lineage sample has the allele frequency of 1 at this SNP, and then the population that we're checking to see if it's hybrid is 0.5. So you can kind of eyeball that and see that it could very well be due to an admixture event. Plug it in and we get a negative 0.25. So that's why we use 500,000 SNPs here, so the power is much higher. So we do that, we get the average across that. It still comes out as a negative. We can be pretty sure that this colony is an admixed colony. So that's how we detect the hybrids. I'll show a picture of the result. So we check each combination. So we have our test population here. We have our two populations that made up that colony. So we check all the different combinations, and the ones that are less than 0, we can say that is hybrid. So up here, this is a colony we have here at NYG, and it's a mixture event between this Carnicus sample and a pure Irish sample. That makes sense? So looking at the colonies that we've sampled so far in Ireland, around 15% of them seem to be hybrid. So it's much greater than the 6% we saw in the survey. 7 managed colonies, but 11% for managed population, so beekeepers. And then the free living colonies that we look at, coming in at about 18%. So it seems that beekeepers are helping conservation efforts, which is a great thing to see also. So we can find hybrid colonies using genomics. The next thing we want to know is how hybridized are they? So what's the degree of hybridization? There's another F statistic to do that, and it's called F4, ratio test. So we assume the phylogeny here, so A and B are our pure Irish colonies. C is our intergressing colonies, the C lineage colonies. And O, we need an outgroup to compare it against. So O, in this case, we use the Asian honeybee, Apis cerana. So there's no chance that Apis cerana is mixed with the C lineage or the M lineage. So it's an outgroup. And then X then is the hybrid population that we identified through F3. So just plug the allele frequencies in here, and the ratio ranged from 0.28, so very hybridized, right the way up to 0.93, so not very hybridized. So there seems to be this range in play. So we're able to find hybrids, we're able to see how hybridized they are. How does that look in a population model? So to do this, we took our Irish data set, we added it to an existing Swiss data set and also a European-wide data set. So 3 data sets in together, computed the allele frequencies, and then we did a k-nearest neighbors machine learning classification. And that's the plot we get over here. So if we look at the groups here and how they group, you see the Irish samples group on their own, but then the hybrids kind of moving towards a more continental European cluster. So this is kind of one very strong argument for conservation. We need to stop this from really happening so we can keep our Irish distinct groups. C lineage here, much closer to the hybrids. The Swiss data set, this is from a conservation animal selection area. They show signs of hybridization when you look at the genome. So the Irish data set that we have is one of the last real pure holdouts for the eminence. So conservation should be done to keep this going. And the last thing I'm doing is the microbiome analysis. So when we do the DNA extraction, we take some tissue from the abdomen. So we map all the reads to the honeybee genome. Anything that's left over, we kind of call it the microbiome. And I wanted to know, does management in any way kind of influence the microbiome? And the initial results seem to say that it does. So we did a pairwise Adonis test, p-value significant. We did a Shannon diversity showing the diversity in the microbiome. Both seem to be different. So we took that result and we looked at where exactly is this difference coming from. So we use DSeq2. This is using RNA-seq a lot, but it's just a program that compares counts of things to see if there's any significant difference. So I compared the counts of each bacteria between wild and managed, and we see a lot of upregulation and a lot of downregulation. So there's definite difference in the counts of bacteria and the microbiomes. So now that we know that there's a difference in the Shannon diversity, we know there's a difference in the actual counts. We went on to see, does this difference in bacteria counts go on and change the differences in pathways? So you can, each microorganism will contribute to a pathway, so they take food in and they give something out. So this is microorganisms that it gives out L-lysine. And we see a significant difference between our managed and our free living for this pathway. There's many different pathways like this. I just started looking into this, and you can see here the relative abundance. There's much different. There's differences in the lactobacillus here. So that's where we're at the microbiome stuff. If anyone wants to help out in that, I'd be very grateful. So yes, a summary, we were able to find hybrids. We'll see how hybridized they are. We can see that hybrid colonies move away from the Irish cluster into more European. So conservation is definitely needed there to make sure that stops happening. And management seems to create a specific type of microbiome composition compared to free living. So thanks for everyone for listening, for coming. And thanks to the lab for helping out. Thanks to SS5 for the funding, of course. And any questions?
Thanks, Stephen, for playing on time as well.
I'm just going to share it with people.
I'll open up the floor to any questions. Where's the chat?
Yeah, just coming in.
Darren, you have a question there? All right.
Yeah. Hi, Stephen. Really great talk. Very interesting. I found it's very interesting that you get an 18% hybridized wild colonies, and then obviously you've got a lower number for the managed. Mm-hmm. Do you think that's self-reporting, basically? So from my own sampling, I found people who didn't have native Irish bees were more reluctant to engage often because they're afraid that we'd sequence their bees and somehow highlight that to NIDS. So is the wild level of hybridization probably a more realistic level for Ireland, would you say? Are you more likely to be getting pure Irish bees from managed populations because people who have them are more likely to want them to be sequenced?
Yeah, that's definitely a good point. The survey that I kind of touched on at the start, we asked them various different questions, and one of them was, are you willing to participate in research? And when you look at the responses, the people who keep non-native bees were very reluctant to participate in research. For all the points that you pointed out, they are seem to be villainized in Ireland for keep. So yeah, they do keep them themselves. So that is a very good point, and it could very well be the case, but there's no way to know that until they want to participate in research. Really.
Yeah, and I just want to check out, the Swiss bees are M lineage, right, are they?
Yeah.
Okay, cool. All right, I just missed that. All right, thanks very much. Really interesting talk.
Cheers, thanks.
Yeah, very nice talk, thank you. Just a question, so maybe I missed it, but how did you define, like, as your baseline, what was a living literate?
Yeah, that's a good question too. So we use, you'll hear about morphometrics in a bit from Alex, I think, and we're doing the F3 ratio test. So we got reference samples from Carnica, and the European data set has reference samples for Ligustica. So
did the Carnica and Ligustica examples come from Europe?
Yeah. So we were able to use the F3 statistics to see if they're any way similar to C lineage. So it's kind of, we have around 200 samples, so it's kind of just a lot of permutations to find out which cluster is actually M lineage, which samples are actually M lineage using morphometrics, reports, then the actual DNA. Yeah.
Mm-hmm.
Yeah.
So you did do morphometrics on all the samples as well?
Yeah, Alex will touch on.
Thank you very much, that was really interesting. And I just wondered whether you'd had any chats with Sarah Larragee in Jim Cameron's group in Maynooth, because she's doing something really similar with Bombus terrestris.
Not happening. Sarah
Larragee. She's not here, she's great enough at thesis as well.
Okay. Then there's population stuff?
It's genomics and microbiomes and stuff. Yeah. I was just thinking she just sent me a chapter to read on the microbiome stuff. So.
Oh, that'd be cool to know, because this is all new to me, the
so
microbiome stuff.
make that link between the honeybees and the
that'll be great, yeah.
Because there's the same issues with wild and commercial. And
they're different also in honey, they're in bubbles?
British and European,
Maybe I'm on the right track.
Just one quick question with the microbiome. What was your method then? You sequence the whole bee, did you?
So what we do is we get the sample in, we freeze dry it.
So that's the bee?
The bees, yeah.
Yeah.
We get the bees in, we freeze dry them, and then we extract only the thoracic muscle, and then we scoop out the abdomen and add that also. So we have the abdomen and then the thoracic muscle. That's what we use for microbiome.
And then just whole shutdown sequence?
Yep, whole genome sequencing, yeah. So you can we're really looking at just the abundances that are in there. So it's different than 16S and stuff.
So then you're taking away, mapping to the honeybee genome to take away the honeybee.
Yep, and then I also map to the human to get rid of the human stuff, and then whatever that.
Human stuff?
Just in case if there's human got in there when we were doing the dissection and stuff, just in case. So yeah, we just left it. What should be only, because we use the thoracic muscle only, and should be whatever's in that should be shown up as that.
Yeah.
That's the idea.
Yeah, right. Very good.
Okay, I'm going to move things on because
I'm gonna go.
10 seconds. Thanks very much, Steve.
Thanks very much.